So good afternoon, everybody, and welcome to your third class in biology. And today we're going to begin talking about the structure and function of large biological molecules. There are some small biological molecules that we will encounter, but the vast majority of biological molecules produced by the metabolism of the cells during anabolic processes are in fact very large. Most of them are extremely large. And they obviously all have foundation skeleton made of carbon atoms which are joined together. When we join carbon atoms together with covalent bonds, those bonds are very, very strong. And that enables very large molecules to be built. And when we start adding functional groups and different elements to that carbon skeleton one way or another, we begin to change the nature of the molecule. So living things will create molecules which are needed in the cell to perform particular functions. And what we find is that their structure is always intimately connected to their function. And we'll see this again and again and again. Even slight changes in the composition of a molecule, slight changes in its structure can result in radical changes in its properties and in its functions within the cell. So the first thing that we're going to cover are two groups, the carbohydrates and the lipids. And both of these very large molecules, but quite different to one another, as you will see. So we actually recognize four groups, by and large, of biological molecules, or biomolecules. And these are the carbohydrates, lipids that we'll do today, and then the proteins and the nucleic acids. When we see that word, that term, macromolecule, it's actually rather a loose term. It simply means a very large molecule, which most biological molecules are. There's no lower limit where something stops being a macromolecule, but so it's somewhat vague in that sense. But most of the molecules that we deal with in living systems are macromolecules. They're extremely large molecules. And we're going to discover that for all of them, it is that foundation, if there's a foundation sequence to these molecules, which confers their basic properties to them. And that areas of the molecule may be changed in one way or another to confer other biological properties. So for example, if I was to show you a protein can be extremely long. A protein is composed of amino acids, a whole lot of amino acids all joined together in a long chain. But you remember that the amino acids you must remember that the amino acids each have different properties, and I can gather amino acids together with similar properties in one area of a molecule and confer that nature to the molecule. For example, there are hydrophobic amino acids. If I start gathering a whole lot of hydrophobic amino acids together in one region of a protein molecule, I'll make that whole region of the molecule hydrophobic, and I'll get it to interact that way with water that surrounds it. It will try to be repelled by the water. I may be able to start folding my protein molecule like that. I can confer particular properties to particular areas of a molecule by concentrating functional groups there. But the basis of the molecules are very similar so that they can be joined together. This will become clear in a second. Because most biomolecules are what we refer to as polymers. And a polymer is specifically a molecule which is made from many similar repeating building blocks, which are joined together. And those units are called the monomers. We have three classes of our biomolecules, which are polymers. And those are the carbohydrates, the proteins, and the nucleic acids. So those three are all polymeric They are polymers formed from monomers. The lipids are not polymers. They're very large molecules. They're macromolecules, but they're not polymers. They don't have monomers. There are no lipid monomers to make lipid polymers. But carbohydrates, proteins, and nucleic acids all composed of monomers joined together to form the mature molecule. Carbohydrates, the units which are joined together are simple sugars, monosaccharides. Proteins, the monomers of proteins are amino acids. Amino acids joined together will end up forming a molecule which will ultimately form a protein. Nucleic acids are formed from monomers called nucleotides. And DNA, for example, is an enormously long molecule, and it's composed of a series of nucleotides all joined together. Again, lipids are not polymers. They are macromolecules, but not polymers. So we can form these polymers in a specific fashion. Here, for example, are monomers which are being joined together. We've already got 3 joined together here in a chain, 1, 2, 3. And here is a 4th, which we want to join to this chain. And we do it in a specific way. We do it by removing from the molecules a molecule of water. There it goes there. That's HOH. That is H2O. This is common to the formation of all polymers. They are formed by a dehydration reaction. HOH, out it goes. That is dehydration. We're removing water, and we're forming a new bond here and joining monomer 4 to the already existing chain. So that's quite simple to understand, but there's some really important implications in what we've just seen. The first is this system works because all the monomers have a portion of them which is extremely similar, if not identical, in all the other monomers. Okay, so this, what we're looking at joining here are the identical portions of the monomers. The monomers may be very different to one another. Let's think, for example, if this was a protein and these were amino acids. Well, we can have amino acids which are very, very hydrophilic, amino acids which are very hydrophobic, amino acids which are polar. All sorts of different properties are associated with amino acids, but they're not associated with their core, with this root part of the amino. All the amino acids, no matter how different they are, they have one part of them which is identical. And that means that any amino acid can be joined to any other amino acid. The part that gives the amino acid its character is off as a side branch here, and it doesn't participate in the polymerization. It doesn't participate in the chain formation. The chain formation occurs because all amino acids have an identical part. So does this, okay? The one that we joined in here, also identical here. And that's the really significant thing about this is I can join any monomer to any other monomer. Because they're being joined through identical parts, then they're not being joined through their differences, they're being joined through their similarities. It means I can join any monomer to any other monomer. I can establish any order. And I can do it with very few monomers. I only need a few monomers and I can build molecules of immense diversity because I can put those few monomers together in any order. That's not to say the order that they're put together in is random. It is not. The order of the monomers being put is determined by the cell, and it's ultimately determined by the DNA. That is the role of DNA. DNA contains the recipe for all of these molecules. And DNA contains, in polymers, DNA ultimately contains the recipe. For example, for the proteins, DNA contains a recipe for all of the amino acids. It is DNA which determines that amino acid sequence in proteins. Now, so through that process of joining, we can form very, very long polymers. thousands and thousands of units of monomers long if need be. But the cell doesn't only build, it also breaks down. And it can break down these large molecules for a variety of reasons. It can break them down, for example, for structural reasons. It may want to break down a molecule and change it to something else. It may want to break down the molecule to extract the energy that is contained in this bond. All of these occur, so we have to be able to break as well as build. These building reactions we call the anabolic reactions. They are the metabolic activity which builds biomolecules. Metabolic activity which breaks down is called catabolic. And it, as I say, it happens for a variety of reasons. Now, when we want to break down a polymer, we do so by adding water back into the bond. So here's a water molecule. We put in H2O. So we put in water, and because of that, we call this an hydrolysis reaction. It is a hydrolysis reaction. Dehydration to build, hydrolysis to break down these polymers. So that's the basic mechanisms, and the important takeaway was for these polymers, we can put the monomers together in any order. We only need a few monomers to build molecules of immense diversity. So for example, all of our proteins are built from just 20 monomers, which are the amino acids. There are only 20 amino acids used in the living cell. And all the nucleic acids, which have to be incredibly diverse, we build from just 5 nucleotides. Just 5 different nucleotides make up, and we make up all of our nucleic acids. The nucleic acids are DNA, deoxyribonucleic acid, and RNA, ribonucleic acids. And again, the DNA has to be, first of all, it has to be very, very big. And it has to be able to be very diverse, because it's got to contain all of the recipes for everything that happens in the cell. In fact, it contains the recipes for all of the proteins that are in the cell. And there are thousands and thousands of them. So DNA contains a huge amount of information. That information is locked in to the sequence of its monomers, to the sequence of its nucleotides. That is what carries the recipe for the proteins. And that recipe for a protein we call a gene. And the nature of the gene is locked in to its sequence of nucleotides. Let's have a look at the first group. These are carbohydrates. And carbohydrates in general are sugars and the polymers of sugars. And there are some alcohols as well that we would consider as being sugars. But the very simplest carbohydrates are the monosaccharides. They are single sugar units. And the monosaccharides are the monomers of carbohydrate polymers. And the carbohydrate polymer is called a polysaccharide. So we're going to see that we can take single monosaccharide units and we can join them together. And we'll end up producing molecules whose nature is entirely different to the nature of the monomers that make them up. The macromolecule, carbohydrate macromolecules, are polysaccharides, many sugar units. Polysaccharide, many sugar units. And the sugar units are simple sugars or monosaccharides. And the monosaccharides usually have this formula. It is CH₂O. We'll look at it and you'll understand where the name carbohydrate comes from, because it's carbon joined to water. It's hydrated carbon, and that gives you carbohydrate. In the biological systems, the commonest monosaccharide is glucose. And there's an important reason for that. Glucose is the first molecule which is produced from the process of photosynthesis. And plants take carbon dioxide and they take water. And they use the sun's energy to build bonds between those atoms to form this molecule, C6H12O6. Have a look here and you'll see, let's divide these by 6, CH₂O, CH₂O. There it conforms to that formula, CH₂O. It's a very, very common monosaccharide in both animal and plant systems. It's one of our principal sources of energy, especially quick energy. And we'll learn something about it, how it is metabolized, everything else. Because it's a very significant molecule. It makes its appearance very often in metabolic systems, for example. So in solution, glucose is not a carbon chain. When you crystallize it out of solution, then it may be more linear. But in solution, it tends to form these elaborate rings. And I just want you to get used to having a look at this. This is a sort of shorthand for molecular structure, because at each of these points here, not there where an oxygen is drawn in for you, but at these points here where there is nothing indicated, there is a carbon. And these carbons are numbered. C1, 2, 3, 4, 5, and here is C6. It has 6 carbons. And at each of these carbons, there is HOH, HOH, etc., all the way around to give you the final form. C6H12O6, please remember this. Okay, C6H12O6 is glucose. And this glucose, in actual fact, and similar monosaccharides are the monomers of much larger molecules that can be built from them. And if we have a look here, they are built in exactly the way that we described before. Have a look, and we see here's a glucose molecule here, here is another one. I can join these together, and I join them with a dehydration reaction. HOH, take that out, it's dehydrated, and we join them together. And this bond here, you must remember, is a glycosidic linkage. It is a glycosidic linkage. Don't worry over much about the numbering. The numbering is where the bonds take place. It takes place between C1 and C4. That's a 1,4 glycosidic linkage. I just want you to remember there's a glycosidic linkage. And it forms this molecule here. This is another sugar, which we commonly encounter, especially in microbial systems. It is commonly produced from fermentation by microbes, and it is used by microbes. And it's a sugar called maltose. And it is the same sugar that gives malted products their flavor. It's used for everything from malted milk drinks, malted ice cream, all the way up to malted whiskeys. And it is usually produced industrially by fermenting grain. Grain is dampened, allowed to sprout, and it is then crushed and allowed to ferment, and the microbes associated with that produce rich amounts of maltose. And here it is here, 2 glucose units. Because it's 2, this is called a disaccharide. 2 sugars, disaccharide. If we added 3, it'd be a trisaccharide, but anything over that, we'd probably call it a polysaccharide. So the 2 units, the 2 monomers, do not have to both be glucose. They can be different monosaccharides. The monosaccharides are mostly so similar to one another that we can perform this dehydration reaction between them. And here is one here. This is glucose. This is another plant sugar called fructose. Fructose is commonly produced by plants. Some plants produce more than others. And for example, corn produces a tremendous amount of fructose. And that's where high fructose corn syrup comes from. It is produced by corn. And here's fructose. Here it's very, very similar to glucose. There's one bond less in the ring, and instead it has a branch at the side here, in addition to this branch here. But otherwise, it's very, very similar to glucose. Here's glucose here. Here's fructose. Fructose's properties are different to glucose. We can still join them together in a dehydration reaction, HOH here. And there goes the water. The dehydration causes a glycosidic linkage here. And we have another disaccharide called sucrose. And sucrose is ordinary table sugar. That's the sugar that we use most commonly. Just remember, it's a glucose molecule together with a fructose molecule, and it is a disaccharide with a glycosidic linkage. We don't have to, as I've said, we don't have to stick at 2. We can go 3 and up, and very frequently do. So when we do so, we call these polysaccharides. And both the structure and the function of the polysaccharides is dependent on, first of all, the monomers, which are used to make them, most commonly glucose. Then the orientation of the monomers. The monomers can come in different orientations, and the nature of the bonds between the monomers. All of these things affect the architecture of the molecule and its function. And we'll see that they often have very, very, very different properties indeed. The most commonly encountered in plants, there are 2 polysaccharides, but as a process of photosynthesis, the process of photosynthesis takes place in a chloroplast, and this is a chloroplast shown here in the plant. The chloroplast rapidly makes glucose, and when it makes glucose in excess, more than the plant needs, it will immediately start to convert that glucose into a storage polysaccharide called starch. And that is typically a plant product. We do not get starches in animals. We only get it in plants. Starch is a very long molecule, and it is insoluble. And so it begins to precipitate out and crystallize. And it will form these distinct white granules inside the chloroplast. Eventually, chloroplasts become packed with the starch like that, and they stop functioning as chloroplasts. Doesn't hurt the plant because the plant produces millions of chloroplasts the whole time. They're produced by binary fission, so they always have a big population of active chloroplasts, but they use them to store starch. Other plants will store starch granules inside specialized cells. For example, in tubers like potatoes, there are specialized cells which are jam-packed with starch granules. So that is the chief storage product of plants is starch. And we have a look here, and we'll see there are different forms of starch inside a plant. The simplest is called amylose, and it's a straight chain of glucose units, all joined together by glycosidic linkages. And if you have a look here, in actual fact, you'll see that these starch units in glucose, they're all in the same orientation. Okay, they're all in the same orientation, exactly. And then starch can also be branched. That's a substance called amylopectin. You don't need to worry. Just remember that these all form starch. And despite the fact glucose is highly soluble, it forms such long molecules that it becomes insoluble and precipitates out. Now, there is a very, very similar substance in animals. It is not starch, though. It is called glycogen, and that is our chief storage product for intermediate energy storage. Our long-term storage is fats and lipids, but our chief intermediate storage product is glycogen. And glycogen is a substance, again, it is made from it is a long polysaccharide of glucose monomers. But now the units are again in the same orientation, just like starch was, but it is branched. It is highly branched, and just that alone confers different properties. And glycogen occurs only in animals, and it is laid down in largely in the liver as a store of glucose units, and in the muscles. In the muscles, because muscles require a source of fuel, and they may need it very rapidly, rather than having to mobilize glycogen from the liver and transfer the glucose units to the muscles. They have their own glycogen stores, which can be accessed if they need energy. The construction of glucose into glycogen molecules, the manufacture of glycogen from glucose, is governed ultimately by a hormone called insulin, which governs the passage of glucose into the cell. And there are other hormones which then take the glucose and put it into the glycogen. And glycogen is the storage product. When we need glucose, we break down glycogen and liberate glucose. When we don't need the glucose, we take glucose out of our bloodstream, we put it into cells, and we create glycogen. We make, in fact, create so much glycogen that it actually precipitates out, is visible inside the cells. Glycogen, by the way, is also made in microbes, in bacteria, and is visible inside bacterial cells as well. But remember, starch for plants, that's the storage product. Glycogen, very similar but highly branched. Glycogen is the animal storage product, which is a glucose polysaccharide. Now, the other really important glucose polysaccharide is cellulose. And cellulose is also a plant product. Cellulose is a completely different substance. It is a structural polysaccharide. And it is made from glucose. Again, it's still glucose monomers, one after the other, but now the orientation is different. They are flipped, one after the other. Their orientation is different. Have a look here, and you'll see that the orientation is different. You don't need to remember the orientation. Just remember that the orientation is flipped. The orientation is different in glucose, in starch, glycogen, and then in cellulose. The orientation of the glucose monomers is different, and that makes glucose a radically different substance. It is largely insoluble. And it forms these extremely strong, very, very, very long-lived fibrils, microfibrils, which get packed together, and they form the primary cell walls of plant cells. Animal cells do not have a cell wall around them, but plant cells do. And the initial cell wall that's put around plant cells is made of cellulose. It is extremely strong, and it's what actually is used to give the plant its strength. It does act as a protection of the cells, but it also provides the initial strength for a plant. As a plant grows, it'll put all sorts of other substances into the cell wall to make wood and stuff like that. But the initial cell wall of plants is cellulose. And there is a huge amount of cellulose manufactured by plants. A huge amount of carbon dioxide from the atmosphere is taken in by plants and converted into cellulose as the plant grows. Now, this creates something of an ecological problem, because cellulose, unlike glycogen and starch, cellulose is extremely difficult to digest. There are very, very few enzymes which will attack cellulose, and animals do not possess them. Animals like cows, us, etc., do not possess them. So I'm sorry, I forgot that I have this diagram here. Here you can see the difference. Here's starch, or it'd be the same for glycogen. Okay, so here's the orientation of starch. Here is the orientation for cellulose, and you can see that the units are flipped as you go down. So we do not possess any enzymes, nor does any other mammal, which can break down cellulose. Now, this creates a huge ecological problem, because if plants are putting in so many carbon molecules into cellulose, building it into cellulose, if cellulose is never broken down, it would end up sopping up all of the carbon on the planet eventually, in all of the billions of years that photosynthesis has been going on, etc. Eventually, all the carbon molecules would have got locked up into cellulose. Cellulose is very, very difficult to break down. In us, when we eat cellulose, it passes through our gut untouched. We don't digest it at all, and it's what we call fiber. That is dietary fiber. Fortunately, there are many microbes, both bacteria and some fungi, and also a few protozoa, which are able to break down cellulose. And all of the degradation of cellulose, which is an essential part of ecological cycling of carbon, that is all undertaken by microbes. And then you have to ask the question, well, what about animals, for example, like cows, or cows, termites, all of these things? They depend almost entirely on plant material. And the bulk of what they eat is actually cellulose. It would be somehow very wasteful if they just simply voided all their cellulose as dietary fiber. In fact, it wouldn't work for them. They can't get enough nutrient out of plant cells on their own to survive. They have to have systems to digest the cellulose. And what these animals do is they have dedicated areas of the gut. Their guts are specialized to house microbes, which will in fact digest cellulose. And then some of the product is given to the host animal. So this is a symbiosis, a symbiotic relationship. In animals like cows, the stomach is extensively developed into different regions where there are different microbial populations which process the cellulose. And then the animal is able to absorb products produced as a result of that digestion. Even animals like rabbits, for example, have specialized guts and specialized feeding habits, etc., in order to digest the cellulose, make use of cellulose instead of having it just pass straight through the gut. Remember this word. Those are symbiotic relationships. Symbiosis means living together. And in this case, that symbiosis is mutually beneficial, and it's called a mutualism. Both of them benefit. The microbes benefit. They've got a warm, safe habitat with a regular supply of food. The host benefits because it gets products from the cellulose digestion. We all benefit because cellulose has to be broken down into smaller units, which can then be used by other organisms. Otherwise, all carbon would end up being tied up in cellulose. I'll just make mention of one other important polysaccharide, and this is another structural polysaccharide, and this is a substance called chitin. And here's a chitin monomer, and chitin is a little bit different because it has a sugar unit here, and then it has a nitrogen-rich unit here attached to it. Okay, so it's a little bit different. Chitin is another structural polysaccharide, very, very strong. Again, actually, it's also quite difficult to digest, but there are microbes which will digest it. But chitin is most commonly found as the exoskeleton of arthropods, of the animals called the arthropods. This is chitin here, and it has a chitinous layer all around the outside. Chitin is also present in fungi. Chitin forms the cell walls of fungi. So just remember the name. You don't need to remember the structure there. Just remember it's another polysaccharide. We'll join a whole lot of these. This is a monomer. We join a whole lot of these together to form the chitin itself. Okay, so that's all that for the moment, all we're going to talk about with the carbohydrates, but believe me, we'll hear a lot more about the carbohydrates when we start to talk about metabolism, especially about glucose. Let's have a look now at this group of macromolecules called the lipids. The lipids are the fats and the oils. Again, they are not polymers. They are macromolecules. They can be extremely large, but they do not form polymers. There are no lipid monomers to put together to form lipid polymers. And the one thing which distinguishes them, they're very diverse. We're going to see there's some which look they often look quite unlike one another. They all have this one property in common, and that is they are all strongly hydrophobic. And there are three groups that we're going to talk about. We're going to talk about fat lipids, first of all, as a single group. And then a group called the phospholipids that you've actually already heard about because we talked about them briefly when I told you about the cell membrane. And then the last group we'll just mention are the steroids. These are all very different to one another, but they are all strongly hydrophobic. And that is what classes them as being lipids. So the fats are something that you have obviously heard about. The fats are a storage product largely, especially in animals. They do occur in plants as well, but they are most commonly associated with animals. And the fats have a very specific structure. They have a sugar unit called glycerol. And that unit joins together a group of molecules called fatty acids. Fatty acids are hydrocarbons, and they are called fatty acids, first of all, because of their association with fat, but they can exist on their own as a long molecule with a carboxyl group at the end. And you'll remember that that carboxyl group confers an acidic character to a molecule. So glycerol is actually an alcohol sugar. So it's a three-carbon alcohol sugar, and it can bind at each carbon. It can bind one fatty acid chain. So here we have, and I think if you check through your own PowerPoint presentation, I think there's a little animation for you of this process happening. This is a similar reaction to what happens in polymerization, but remember, this is not a polymer. In that we form the fat by joining the fatty acid to a glycerol molecule by dehydration reaction. Here it is here. We're going to take out that water molecule, and we're going to join this fatty acid to the glycerol. We know it's a fatty acid because look at its structure. It's a long hydrocarbon here, very, very hydrophobic. And it has at the end here a carboxyl group. And that carboxyl group is going to confer an acidic nature to it. So this is a fatty acid. It's a typical fatty acid. There are many fatty acids, many kinds of fatty acids, and many of them will participate in the construction of different fats. So the glycerol can bind up to 3 fatty acids. And when it does so, this here, again, a name that you may well have heard, and that is that this is a triglyceride. This is a triglyceride fat. Triglyceride because it has 3 fatty acids. Those fatty acids don't all have to be the same fatty acids. They can be different fatty acids. And that, depending on what they are, the fat may have different properties. So this is a triglyceride fat that is typical for us, typical for animals in general. And I just want to point out one other thing in saying that the fatty acids can be of a different character. first of all, have a look here, and you'll realize if we look at these carbons here, they have as many hydrogens attached to them as possible. Remember, each carbon has 4 bonds it can make. So it's got 1 bond to the next carbon. So it has 3, the end one has 3 available for hydrogens. They're all filled. Each one down the chain has 2 bonds available for hydrogen. They're all filled. In other words, there are no double bonds down here. No double bonds. This is called a saturated fatty acid. And this altogether is a saturated fat. There are as many hydrogens here as possible. And that makes it a saturated fat. And there's another property conferred by that. These are relatively straight, these fatty acids. If I took any one of these hydrogens here off and used that to make a double bond there, I would have 1 less hydrogen. That would be an unsaturated fatty acid. And that double bond there would give a kink, would induce a kink in the fatty acid tail. It would change its shape in 3-dimensional space and thereby change its properties. Remember, we said that before when we looked at carbon skeletons. We can change them by changing their bonding. We can make the double bonding to induce a 3-dimensional change. And then they have 1 less hydrogen than they could have. Saturated fats are most strongly associated with animal products. So they're most strongly associated, and they're usually triglycerides, which can be solid at room temperature. So they would have saturated fatty acids in the fat. Those saturated fats are most strongly associated with animals, and they are also strongly associated with adverse health outcomes. These saturated fats are believed to contribute to all sorts of aspects of human disease, and not least to cardiovascular disease, because they appear to be strong contributors to the formation of plaque inside the blood vessels of the cardiovascular system. Saturated fats are things like cream, butter, cheeses. All of these things are very, very rich in saturated fats. The unsaturated fats are more strongly associated with plants and with plant metabolism. And a typical example would be is olive oil. And you can see here it's liquid at room temperature. Now this room temperature rule is quite a vague one. It's not really a strict definition. But if you go to the grocery store and you go to the olive oil counters, if the grocery store has their air conditioning turned a little bit too low, you'll see that the olive oil itself will go solid. There's nothing wrong with it. You just take it home, warm it up slightly, and it becomes liquid like this. And that's typical for an unsaturated lipid, unsaturated fat. Here is the structure of fats from olive oil. And you can see here's the glycerol molecule again. Here are some fatty acid chains, but this one here is unsaturated. Look, it's got a double bond in it, and it causes the molecule to be bent in 3-dimensional space. And that alone is enough to confer all sorts of different properties to that fat. And these fats and oils are associated with better health outcomes. These are regarded as being far more healthy part of the diet than the saturated fats. So there is a third kind of fat that we need to talk about. Nowadays, less important than it used to be. These are called the hydrogenated fats. They are artificially produced fats where they took saturated fats and they kind of turned them into artificially unsaturated in a way. And these were very, very advantageous for industry. These fats and oils that we're talking about here, they have a disadvantage. That is that they oxidize quite easily once they leave the cell and they are put into an oxygen-rich environment. They tend to oxidize quite easily. And when they do, they end up breaking up these fatty acids. The fatty acids get split off, and the fatty acids on their own are very unpleasant tasting. This is the taste of rancidity. If butter goes rancid, what has happened is its fatty acid chains have been broken by oxidation, and you accumulate these acidic fatty acids, which really do not taste good at all. This happens quite rapidly. In the old days, when all they had was saturated and unsaturated natural products, rancidity was a tremendous problem. They would include preservatives of some sort to try and stop it happening. But then they invented these hydrogenated fats. The classic example is margarine. They look and behave something like butter, and they can be used in all sorts of manufacturing processes, in all sorts of baking processes, and they are not oxidized, especially by microbial action, very quickly. So stuff made with hydrogenated fats like this they are for industry very advantageous because the product is very long-lived. And these were used very widely. The oils were used, for example, for frying chips, for frying donuts, because the oils are very long-lasting. They don't break down under heat. They don't oxidize under heat. Biscuits, cookies, crackers, all these sorts of things made with hydrogenated fats were very long-lived. They didn't go rancid. They didn't go stale. But the problem was they discovered that trans fats, as they're called, are extremely bad for the health. They are associated with extremely poor health outcomes. They are far, far worse than the natural saturated fats for causing cardiovascular disease especially. And their usage now is becoming quite rare, but you should still check. When you buy cookies or anything, just check that they don't have trans fats in them, because they really are not good for you at all. Okay, so those kinds of fats, like we're talking the triglycerides and all the animal fats especially, their principal importance is as a source of energy. And the reason for that is just look at them. All of these carbons all join together with bonds, and all of the carbon with hydrogens associated with them by these bonds. There are just hundreds of bonds in these big molecules. And each of those bonds is a potential source of energy. Each bond was made by joining two atoms together, and they were joined together by putting energy in. That bond was created by putting energy in. It is a strong bond. It is a covalent bond. It contains a lot of potential energy. And if you can sever that bond, you can actually extract that energy. And that is the principal responsibility of these fats, is to act as energy storage. But this is long-term storage. This is not energy storage like glycerol or like starch. Glycerol and starch are very easily accessed for energy. Fats are not. Fats are for long-term storage, and fats are laid down only when times are really rich, when you have an abundance of nutrients. And even your glycogen or starch storage is full. And you can afford to create the fats or the oils and use them as storage. You put them aside for when things become really, really bad. But for us, there is a problem with that, and that is that actually fat is very difficult to access. It's very difficult to break it down once it's been laid down. You actually need to drive the metabolism in special directions, either by starvation or other ways, to access that fat and mobilize it. So once you've laid down fat, it's actually often quite difficult to get rid of it, especially if you're just leading a normal kind of Western life with a normal diet. Difficult to access that fat. It is a reflection of the fact that in nature, that fat was for long-term storage. That was to see an animal over the winter for those times of famine when other food was simply not available. Then they would access their fat and use it and until good times returned again. And then if they had an abundance of nutrient, then they could build up their fat storage again. Fat also has some other properties. It can be used as well as protection. So for instance, some of our organs in the abdomen have fat associated with them, which is there as a protective coating. The kidneys, for example, have a great big fat cap over the top of them. Mammals have specialized tissue to contain fat droplets, and that is called adipose tissue. And so you look at the kidneys, they have two big collections of adipose tissue that cover the top of the kidneys, and that is a protective. In other cases, fat may act as an insulator. Fat is often in Arctic animals, things like that. In marine animals, it is laid down under the skin and it acts as an insulator to protect the internal organs and help keep them warm. And it also in aquatic animals, fat acts, because it is lighter than water, fat actually acts as a buoyancy mechanism. So it can be used for flotation. Okay, so we want to talk now about a specialized group of lipids, and these are called the phospholipids. And the phospholipids are tremendously important. All life actually arose because phospholipids arose, and you'll see why I say that in a minute. So there is phospholipids have a very strange structure. They consist of two fatty acid tails joined to a phosphate group. And they have two fatty acid tails, just as we've seen before. Then they have this complex head to them. And the molecule is actually schizophrenic because the two fatty acid tails are strongly hydrophobic, just as fatty acids always are. But this phosphate head is strongly hydrophilic. So we have two properties in the same molecule. And we're going to see that causes the molecule to behave in a very strange way when it's put into contact with water. I don't need you to remember all of the details of this molecule, but I want you to see, here's the fatty acid chain of the tails. There it is. Here's just in a linear model here. They don't have to be the same. They can be different very often. They are very often. There's one of them is unsaturated. So here's one saturated, one unsaturated fatty acid in this phospholipid. They are joined by a glycerol molecule as before, but instead of having a third fatty acid attached here at the glycerol attaches this complex group here, but all that I need you to remember is this here. This is the phosphate here. This is the phosphate group here. And this whole head, that whole head there, is strongly hydrophilic. It dissolves in water very, very easily. This tail does not. It is strongly hydrophobic. So let's do some thought experiments, okay? I'm going to take it's liquid. the phospholipid. I'm going to take a phospholipid and I'm going to drop it into water. Okay. What do you think happens if I just drop it onto the surface of water? Remember, the hydrophilic heads want to dissolve, the tails want to get away from water. Well, what it's going to do is it's going to dissolve its, let me just use this diagram down here because it'd be useful to add it in. It's going to dissolve its hydrophilic heads in the water, and it's going to have its tails sticking out of the water. So it's going to form a film on the surface of the water. Hydrophilic heads dissolved, tails pointing out to get away from the water. So now I'm going to torture it. I'm going to take that, I'm going to shake it, I'm going to force it underneath. And when I force it underneath in that fashion, there are two possibilities. The one is that it forms a structure like this. This is called a micelle. And the micelle consists of a layer of those phospholipid molecules with the hydrophilic heads all dissolved in the water. Remember, I forced it underneath the water. And then all the hydrophobic tails pointing into the center, because what this does is that it forms a shell with the hydrophilic part dissolved happily in the water, and the hydrophobic part here has excluded all the water molecules. So it's quite happy like that. You can do this, it does happen. And that's called a micelle. But there is another possibility. And that is, if I do this vigorously enough, I can force some water molecules into the center here. And then the molecules have to behave a bit differently. The tails have still got to get away from the water. And what happens then is that it does this. It forms an inner shell. Here, remember, we've got water here. It forms an inner shell with the heads dissolved into the water. The tails are all pointing out. And then it forms an outer shell, again, with the heads dissolved in water, the tails pointing in. And now we have a structure where the inside here excludes all the water, and the heads are happily dissolved in water on both sides, and the tails are isolated from the water. What have we got? This is called a liposome. It is a double layer of phospholipids like this, with an interior part here. And the interior part could contain something. It can contain water, or it can contain other substances. We could even take this liposome and imagine that we're going to make it really big. And it's going to enclose cytoplasm, because that is exactly how the cell membrane is structured. You will remember I told you the cell membrane is a double layer, and it is a double layer of phospholipids. Phospholipids have hydrophilic heads and hydrophobic tails. And the double layer is formed like this. If this here is the identical structure, if we just made that cavity in the middle really big and opened the whole thing out, we'd find that we were looking at something that looked like a cell here that had a bilayer with the hydrophobic tails pointing in, hydrophilic heads on the outside. That is the exact structure of the cell membrane. And that when we look at an electron micrograph, that's exactly what we see. We see this double layer. At the cell membrane, we see this double layer of phospholipids. But that is a very, despite the fact it's only 2 molecules thick, it's a very, very good boundary. It forms a very distinct boundary between the inside and the outside. And it's, for example, there are only certain substances which will pass easily across it. Many substances cannot get across that lipid layer. They can't get past the fatty acids, for example. The cell membrane, however, does not just consist of the phospholipids. It's embedded into the phospholipid layer. We're going to see there'll be many, 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 many proteins which confer the living status onto the cell membrane. But its basic structure is exactly this. It is a double layer of phospholipids, heads in the water, tails pointing to the center. Okay, now the very last set of lipids that I want to discuss today are called the steroids. And when you see what they look like, you'll wonder, why on earth do we think that they are lipids? Well, the steroids are lipids, but they have a completely different structure. They have a carbon skeleton that consists of 4 fused rings, and the base kind of steroid is something you have heard a lot about, I'm sure, before, and that is cholesterol. We always associate cholesterol with poor health outcomes, but in fact, cholesterol is an essential nutrient for human beings and for animals, and it forms the basis of all other steroids are developed from cholesterol. It's true, a high level of cholesterol, particular kinds of cholesterol especially, in the blood can contribute to cardiovascular disease. But cholesterol is an essential nutrient. And here is cholesterol. And this also could be the skeleton of many of the other steroids that we're going to talk about. Could talk about. We're not going to talk about steroids very much until we discuss things like hormones, that sort of thing. So here are those rings, as you can see. The reason that it's classed as a lipid is that the steroids are strongly hydrophobic, very, very strongly hydrophobic, and that classes them into the lipids. When you think about steroids, we'll think first of all about things like cholesterol, but then very importantly are the steroid hormones. And those are things like testosterone or estrogen. These are steroid hormones, and those are strongly associated with reproduction, for example, reproduction and development, all of those sort of things. Many of those are steroid hormones, and when we talk about hormones in animals, many of them will be these steroids. Okay, there's just a couple of other things I want to mention before I finish up. I just want to mention something about the fatty acids. Many of the fatty acids that we require, we can actually manufacture by our own metabolism. We can manufacture them perfectly easily. But there are certain fatty acids which we as human beings are not able to manufacture. But we still need them. And so they are called the essential fatty acids. And there are things like omega-3 fatty acids. And that you may have heard of, because if you ever pay any attention to health supplements and things, you'll often see people telling you you must eat omega-3 fatty acids. Well, omega-3 fatty acids, for example, are essential fatty acids. We can't manufacture them, but other animals can. So, for example, we can get a rich source of omega-3 fatty acids is seafood, fish, shrimp, any of those. They're very, very rich in omega-3s and in other essential fatty acids. Olive oil has some omega-3s as well. So do many grains. And so if, for example, if you're a vegetarian, this is one thing you need to pay attention to, is that you're having sufficient essential fatty acids taken in. You don't have to get them from animal sources. Animal sources are very rich in essential fatty acids, but you can get them from grains as well. So that would be the last thing that I need to tell you in today's lecture. I hope that you all did fine in your quiz. I'm sure you did. And I will see you then on Wednesday. Don't forget, there's a quiz on Wednesday. Oh, no, sorry. What am I talking about? Don't worry about it. I'll just end up here. Okay, bye.